Welcome back to the fourth topic of our channel 2 in Bio Asia Pacific 2020. And in this presentation is very interesting and a lot of us has been waiting for because we'll be discussing about high resolution HLA genotyping with long read sequencing, ladies and gentlemen. And it's our greatest honor and pleasure to have the honorable representative from Julapon Royal Academy, Dr. Sasithorn Chodwutmantri.
Good afternoon, everybody. My name is Sasithorn Chodwutmantri. I'm a researcher at Julapon Royal Academy. Today, I would like to present you our HLA sequencing project, coming with this title today, High Resolution HLA Genotyping with Long Read Sequencing. Let's start. I believe that all of you are very familiar with HLA genotyping, but what about long read? So NGS, everybody has heard of, is sometimes called next-gen. It's short from next generation sequencing. So this is divided into two generation sequencing, second and a third generation sequencing. Second, we all sometimes call it short read sequencing, whereas third generation sequencing, we sometimes call it long read sequencing. So that's one difference between the two. The second generation sequencing can generate about 100 to 600 base pair of the read information per read, but it has very high accuracy. You don't have to correct the error, or you don't really have to think about the sequencing error for this. However, for the third generation sequencing, it is very long. It could generate from 1 kb up to probably 200 kb for some platform. Well, it's used smart technology or single molecule real-time sequencing technology. There are two companies, one is PacBio, the other is Oxford Nanopore. Gives you similar properties of the sequence read, that is long, but has about 15% error rate for the single read. But why am I talking about long read instead of short read today? Because of the unique property of the HLA that is very highly polymorphic. So if you have these two regions as sketched here, one is dense, one is poor with the SNPs. For the dense region, short read sequencing could give you accurate allele information, whereas if you look at the SNP poor region, it is very difficult to decide which read belongs to which allele, right? But if you do the sequencing using the long read can reach over a very, very long region. And here in this case, it just read one allele and the next allele through and through. You don't have to combine reads together. You don't have to think about whether it's right or wrong if you combine these read to what read, yeah? And you can have one read complete information just in one read and can identify right away which allele it is. Yeah, this is theory. That's why we go for long read. And here, if we are talking about long read, let's look at the PacBio versus Oxford Nanopore. The main difference between these two is the library construct, where Oxford Nanopore allows you to get maximum per single molecule just only 2 pass. It cannot really correct the random error because you just have 50-50 chance, right? But if you look at the PacBio, it can give you multiple pass, could be for sure more than 2 pass. And from what we have done, we get at least 20 passes, which can then generate the consensus read or the CCS read from the original read, and the errors are corrected up to, we will get about 99.9% accuracy for the CCS read. And this is what we use for our project. So what we aim here for our project is the proof of concept of using long read to get the high resolution HLA genotypes, also with face information or allele 1, allele 2, how many alleles there are. And also we aim to sequence 1,000 Asian samples. We want to discover novel alleles in our populations. We want also to contribute these data to building the population level HLA database, which will allow more accurate imputation in the future. And we also would like to discover the relationship of the HLA alleles to the phenotypes in our cohorts. And then we will establish a data analysis pipeline for long-read sequencing. So the design of the project is very simple. So we start with sample collections from our partners. We extract the genomic DNA out. We do the multiplex PCR amplification of class 1 and class 2 HLA genes. We have the full-length class 1, whereas we just have the partial for the class 2 using 1 lambda next type kit. Yeah. And then we prepare these amplicon to be sequencing ready. We call it library preparation, where we add barcodes and we also add the adapter to the amplicons. And then we do the pooling of the libraries, and we found out that we can pool about 96 samples together and then sequence in one run on the smart cell on the PacBio machine, which will take about 10 hours. Then what we get here, we use SmartLink. This is the software provided by PacBio to generate high accuracy reads, which is called CCS, or the consensus read. And we also generate the long amplicon analysis read, which is even more accurate than the CCS read with the SmartLink. And then we use mainly the LAA read to do further data analysis using open source tools as well as some customized pipelines. From here, we have to validate our results, and then sometimes we need to repeat the experiment. And then we have to optimize our pipeline. And in the end, we hope to establish the database and also write some reports. So here just some sketch that I got from the 1 lambda. This is not exactly next type amplicons, but you see what it is. So we have 3 genes from class 1 full length from 5 to 3' UTRs, A, B, and C. Whereas we have the partial portions of the genes amplified for class 2, which we have the RB1, the QB1, and the PB1, or in total 6 genes. Yeah, let's talk about genes. Here, just some details about the library construct with PacBios. So you have double-stranded insert here, and then I like it, the adapter which has barcode here. This is called Smart Bell, which has a loop. And then the polymerase can displace this strand and then sequence in circular through and through. You get very, very long single strand newly synthesized DNA, which has the information of 15% error rate. This is called sub-read. And if you put the sub-reads together from 1 molecules, you can correct the random errors and get the consensus sequence or CCS read, which can go up to 99.9% accuracy. And then you can use the CCS read to generate the LAA read. Here, so that you see the picture, the CCS read can have 1 CCS read per 1 molecule theoretically. So if you have 1 molecule sequence, you will then get out about 1 CCS read with 99.9% accuracy. But one amplicon that you put in the pool has more than one molecules, right? So the next step to generate LAA, the CCS that comes from the same amplicon molecules will be combined or clustered into one group, and then in the end, just only one sequence will be generated. One LAA sequence will be generated per one unique amplicon sequence. Yeah, so we will have about one LAA per one allele per person. This is theoretically. So the pipeline for to analyze these data, this is kind of simple to look at, but inside is very complicated, as I found out. So we start with the LAA reads, which will have just about 12 LAA reads per person. This is very small data set, right? And then in one pipeline, I aligned it against the GRCH38 and also then put it to HLA LA open source software to get the HLA typing. Another pipeline is that I just align it against the HLA sequence that I get from IMGT and then get the typing result using the customized script. Both of them, the results are compared to confirm each other or go against each other. And then when I found out any issues, I go back to the script and have to optimize the parameters of the software to get better results. This is still ongoing and we are still optimizing the pipeline to get more accurate results. So just to see the picture, the gene coverage. This, as an example, is HLA genes. I put the LAA as a query here. So it goes from exon 1 and covered until exon 9. So it's full length for class 1. And class 2, which is not shown here, is just partially covered on the genes. Yeah. But then you see these little red bars indicates the variations in the QRA compared to the reference genome. So the result, at the moment, we have analyzed about 400 samples, and what we found out, this is just an example of the comparison of the results using the HLA-LA software and our customized pipeline. Most of the time, they match to the 6th digit or the 3rd field, which is no problem. But then if you look at the 4th field or the 8th digit, this is always varied, the results. And if you look here, the HLA-LA will give out the G. This means that the 4th field, well, it doesn't mean anything, but it's just that our sequence read doesn't cover really completely full length of some alleles, and not all alleles have the same length, if you can imagine that. Yeah. And this is where we also see our issues with the pipelines and also with the scripts that filter the results. You see some very funny numbering here compared to the HLA-LA results. And when we look closely at the result, at the sequence itself, we found out that this is the problem of the coverage and also the variation in the sequence, and also the mapping algorithm as well. And these are all the issues that we have to fix and optimize to get better results. So From the total of 417 samples that we have analyzed, if I divide it into HLA genes, which are these 6 genes, there are about 620 to 760 alleles found for each genes, which I put into group. The blue group is the percent of the alleles that match between the two pipelines from 6 digits. The red one match only at 4 digits, and the green one match at only 2 digits. So you see that the majority, when comparing the results between the two pipelines, it agrees with each other. Yeah. And here, this graph shows the detected alleles per HLA genes. Here are shown in percent. This is per HLA gene per person in percent. Is it homogeneous or is it heterogeneous? And can we really trust these results? So let's see the results. The blue one gives you the percent of the samples which had 2 alleles detected per genes, normal, right? And the red one has just only 1 allele detected, and the green one has nothing detected. This could be the problem with the samples quality, and also we also have problem with the amplification kit. The stability, sometimes it gives positive, sometimes negative, which we are working on it to solve the problem. So mostly the amplification stability problem occur in class 2 genes, and the poor sample quality affect the outcome, literally. We have no results for the reason of poor sample quality or amplification instability from 5%, around 5% for class 1, and around 5 to 20% for class 2. This is quite a lot, yeah. But we do know that class 2 is more difficult than class 1 to amplify. And these are the top 5 detected HLA genotypes for each genes that we found out among our 417 samples. And if you look at the number here, which I calculate into percent for each genes, so you see noticeable high percentage for HLA genes, which has some alleles with very, very high frequency. Yeah, I don't know the importance of this, but probably, and we will investigate further into this. For the next, I would just like to talk about the issues of nomenclature and the hidden rules of this nomenclature and assigning genotype, assigning typing of the HLA. So if you want to just give 4-digit typing results, for class 1 you just look at 2 exon 2 and 3, and class 2 just look at 1 exon 2. At the same time, if you want to give results for 6 digits, you have to look at all exons. And if you want to give the results at 8 digits, you have to look at full length, including UTR. But this is very tricky. After talking with Charles Core, this is very tricky because the database, the HLA sequence in the database, even from the same gene, doesn't cover the same area or the same area length for each gene. So this is very, very problematic if you want to have a clean-cut genotyping pipeline. I hope this will improve very, very soon. Otherwise, we have to live with it. Yeah. The next issue, which is very important, is that is the mapping selection or filtering algorithm. This is an example of one HLA gene. For example, this is just an example. Yeah. Theoretically, if I have 3 allele in rate, allele 1, 2, and 3, and this is my query. My query goes from this area of UTR to this area of 3' UTR, covering all exon, covering all intron. But if I have these 3 alleles which cover at different lengths of the HLA genes, and I found out, okay, exon 2 and 3, it matched. For this allele 2, it matched the whole length. For this allele 3, it also matched the whole length where my query covered this. But how can I decide whether I would choose allele 2 or allele 3? Well, this is the problem that we have to think about and solve before we can establish real routine pipeline. To summarize the result for now, we have sequenced by 417 samples from Thai and other Asian populations. We have found out that HLA genes has the types that are especially more frequently found compared to other genes. And we found out that around 10 to 20% of the samples are found with only one allele, which have to be more inspected in more detail to see if this is really true homogeneous or it's just because of the sum bias from amplification. We also see that long-read PacBio can give about 70 to 90% HLA typing with 6 to 8 digits. And the customized pipeline is also a good alternative, given that it is more optimized to fix complicated issues. And also, we now established that 96 samples can be pulled together and sequenced in one SMART cell. And this can give the sequencing cost per sample around 4,000 to 7,000 baht, depends on different factors. Yeah. And then there are some issues with the amplification stabilities using this 1 lambda kit, which theoretically could be fixed with higher sample quality and higher sample amount. And if we use the LAA sequence reads for further downstream analysis, there is no need at all for high computing power to do the analysis. So you can do the analysis by yourself in your PC, on your PC. But right now, in some cases, CCS read is still necessary to reconfirm the results. So if you are talking about CCS read, you might need a good, maybe better than normal PCs to do the analysis. And then we found out that the complication with the HLA nomenclature and the incomplete or inconsistent sequence information make it very difficult to optimize a clean-cut pipeline. And I hope this will soon change. And I would like to many thanks to our collaborators and teams at Ramatipati Hospital, Professor Vasant's team, and also Professor Cholapat's team. And also Dr. Surakamet's team at the Department of Medical Science at the Ministry of Public Health. And I would like also to thank you, Dr. Pichaya at Julapon Graduate Institute, as well as many thanks to Dr. Charles Kaur. If you have any question, please feel free to send me the question to this email. Thank you.
Thank you very much, Dr. Sasithorn. I am certain that our audience out there have an even better and clearer understanding regarding high-resolution HLA genotyping with long-read sequencing. And without further ado, I would like to happily announce that because some of our participants would like to take part in elaborating even more on the topic. So I would like to introduce to you this next part, which is the Q&A session for the better elaboration on the topic. Would you please?
So thank you for your attention. And for the first question, it was asked, 1,000 Asian samples can see the relationship and HLA typing, right? Well, 1,000 Asian samples, we have to talk about, at the moment, we put the focus on Thai population, which we right now give the quota for Thai populations about 500 samples. So talking with Charles Core, he's an expert and is working in Japan and working on giving better imputation of HLA among Asian populations. So he suggests that we need around 400 samples per each population to see the big picture, to get better imputation for each Asian population. So for our project right now, at the moment, we will have 500 at least samples from Thai. So we can hopefully, theoretically, will see some relationship, at least for Thai population. But for other countries, we are still collecting the number of samples, and hopefully we will have the phase 2 of the project, and we will sequence in a much larger number for other countries. Yeah. Second question, for using long read, how about the cost effectiveness compared to shortener techniques? Well, I haven't really actually compared the cost with the short read sequencing, but as you see in my quite last slide, I estimate that the cost per sample to sequence full-length class 1 and partial class 2, at least for these amplicon using this kit, will cost around 4,000 to 7,000 baht, Thai baht, per person. And this is the cost. We can push it downwards if we pull more samples together, which I think is still possible to pull up to quite almost 200 samples per smart run. So the cost will go downward, but this have to be optimized further. However, I think we still right now have a bigger problem other than the cost, because I think the cost, it shouldn't be any problem. The other problem is that we are still working on the stability of the amplification. And I think if we are allowed to use more sample amount, and if the sample amount, if the sample quality is very good, then I think most of the time we don't have any amplification problem. Yeah. Have any of you any more further question? I would be glad to answer. Otherwise, you can send me an email in the